I moved about 2 years ago to the University of Mainz and established a lab with two missions. On the one hand, we want to explore plant defenses, on the other hand, exploit these defenses. So on the one hand, we want to understand how plants, and in particular duckweeds, protect themselves against environmental stresses, mostly herbivores, by producing different bioactive molecules. On the other hand, we want to exploit these duckweeds to turn them into green biofactories to produce the metabolites that we need in our society. Meaning we want to introduce biosynthetic pathways in these plants to make them to produce the metabolites that we need. For today's talk, I would like to mostly focus on these more ecological side. And particularly, I would like to present data from one of the PhD students in my lab, Alexandra Chavez. And during her PhD, Alex wanted to assess whether plants' defenses are transgenerational, plastic, and whether they alter plant and herbivore fitness. So phenotypic plasticity, and in particular transgenerational phenotypic plasticity, indeed may become more and more important during global change. Because on the one hand, we have increasingly rapidly changing environmental conditions, and on the other hand, intraspecific diversity is declining, meaning that adaptation through genetic change may simply not be possible or not be fast enough to make plants resistant to the environmental challenges. Plasticity is a very well-known phenomenon in terms of plant defense and the induction of specialized metabolites, at least within generations. What is much less known is whether plants are also phenotypically plastic across generations. And the main hurdle in my eyes has been that it is simply very painful to study transgenerational plasticity, because for most plants it takes months, if not years, simply to finish a single cycle. So if you want to finish your PhD within a reasonable time, and studying transgenerational plasticity may seem to not be the best idea. And as such, we turned our attention to a new model system, the giant duckweed Spirodela polymeriza, which is a very inconspicuous plant. It's just these small green floating leaflets that you may know from certain ponds in nearby. And although the plant is very tiny, or maybe just because the plant is very tiny, it is really exceptionally suitable for transgenerational studies because of two main features. So first of all, it's simply one of the fastest growing and fastest reproducing flowering plants that exist on this planet. It reproduces very quickly asexually through budding with a new vegetative generation every 2 to 3 days and by the On the other hand, Spirodela polymeriza has also an exceptionally stable gene. As I mentioned, the plant reproduces vegetatively, meaning that the offspring is the clone of the mother plant, and it is really a perfect clone because when we assess the mutation rate in this species here on the y-axis, we found the mutation rate is at least 7-fold lower than any other multicellular eukaryote that has been assessed. Meaning that if we see any variation between parent and its offspring, we know that this variation must be due to factors other than the DNA. Spirodela, as most other plants, is subjected to various stresses that may fluctuate. A very important stress for duckweeds is copper excess. So copper is often used as a pesticide or also for other settings in human environments. And this copper is a major pollutant in aquatic ecosystems as it disrupts photosystem 2 and thereby causes oxidative stress. And we previously showed that Spirodela protects itself against copper excess by the production of antioxidants, particularly flavonoids, including anthocyanins, which make these plants here, the lower surface of Spirodela plants, turn red. Of course, the plant is also exposed to herbivores, and one of the major native herbivores is the water lily aphid. we have not a good idea how Spirodela protects itself against the water lily aphids. However, in general, aphids are susceptible to JA-induced defects. So we set out to address the question whether these stresses, copper excess in the water lily aphid, elicit transgenerational plasticity in duckweeds, and whether this transgenerational plasticity is adaptive. How do we do so? So assessing transgenerational plasticity is not as simple. The approach we are taking is that we grow the plant as monoclonal single-descendant lineages. Monoclonal because this avoids any genetic variation, and single-descendant lineages, these are lineages in which always a predefined offspring is taken to propagate the next generation. In this way, we avoid the effect of selection on any pre-existing genetic or epigenetic variant. So we grow these monoclonal single-descendant lineages first under a stress and control conditions for about 5 generations. This is what we call a pre-treatment phase. This is the induction. We then grow the plants as single descendants for 5 generations under control conditions to make sure that these plants have never previously experienced the stress. And after this recovery phase under control conditions, we re-expose the plants to the stress or the control conditions and assess plant fitness by measuring growth rates as well as different phenotypes, particularly the production of specialized metabolites. These fitness assays, they last for 8 days. This corresponds to about 2 to 3 generations. So we wanted to, what we then do is to express this fitness and phenotype as the pre-treatment ratio. This is simply, let's say, the growth rate of the copper compared to the control pre-treated. Meaning that when we look at this pre-treatment ratio of growth rate, any value above 1 means that the plant benefits from the previous copper exposure. Any value below 1 means that it pays a price from this previous stress test. So we then wanted to know whether previous copper stress benefits plant fitness under recurring conditions. To answer this question, we used the setup that I just explained, and we used 56 worldwide distributed genotypes that cover all 4 genetic clusters that exist in this species. We then assessed growth rate and counted the number of genotypes that had either positive or negative pre-treatment ratios of their growth rate. We found that under control conditions, so this scenario here, the plants did not bother much whether they had actually experienced copper stress before. However, once we exposed them to the copper stress, we had quite a number of genotypes that showed large pre-treatment ratios. So some of them had almost 50% higher growth under copper stress when they had experienced this stress before. We, however, also had genotypes that paid a price for this previous stress exposure. To assess whether this is really a heritable trait, this transgenerational stress resistance, we calculated pre-treatment ratio, we calculated protein heritabilities, and found protein heritabilities of between 0.5 to 0.6, which is in the range of what we find for the production of specialized metabolites. So this is certainly a heritable trait. We also reproduced some of these genotypes and found reproducible effects of these genotypes that they either benefited from previous stress exposure. So yes, a previous copper exposure can alter plant fitness, but whether the copper exposure benefits the plant really depends on depends on the genotype and on the environment. We then wanted to go into the physiological mechanisms. So what may actually account for this variation in stress resistance, transgenerational stress resistance? So as I mentioned, copper stress inhibits photosystem 2. So that's why we measured photosystem 2 performance, which can be measured with a value that is called FVFM. This basically gives you how well the photosystem 2 is working in the plants. And we expected that the plants that showed a positive pre-treatment ratio of this photosystem 2 would also have a positive pre-treatment ratio of plant fitness. This was the expectation. And when we measured the photosystem 2 activity here under copper excess, we actually found exactly what we expected. So while we had here a positive correlation under copper excess, this correlation was actually reversed under control conditions, indicating that the protection of photosystem 2 involves certain costs in that. So yes, photosystem 2 protection is likely involved in this transgenerational stress resistance. Thus, we were wondering which metabolites may help the plant to protect photosystem 2. To answer this question, we again analyzed the metabolites in this fitness assay, and we did a targeted screening of different phenylpropanoids. These include hydroxycinnamic acid, flavonoids, including anthocyanins, as well as different amines, phytohormones, and amino acids. So in total, about 50 different substances. We then assessed which of those metabolites are enhanced by the copper pretreatment. So we're here looking at the pretreatment ratio of the different metabolites, and here at the p-value, which means that everything below this p-value of 0.05 here means there's no effect of the pretreatment on the metabolite concentration. Everything with a positive pretreatment ratio and the p-value above 0.05 will have an increased levels of these metabolites, and of course, when we have a negative pretreatment ratio, we have a suppression of these metabolites by the copper pretreatment. What we found is that under control conditions, there's not so much going on. We had one amine, tryptamine, that was likely suppressed by the copper pretreatment. When we did the same under copper environment, we again found this tryptamine and popping up. However, also two other metabolites that look very interesting in our eyes: cyanidin through glycoside and its malonylated derivative. So cyanidins are anthocyanins, and anthocyanins are very well-known antioxidants in plants. As I mentioned, they also turn the lower epidermis of the Eastercoots radish. So we wanted to have a closer look at these cyanidins. How come that they were, their concentration was increased upon copper pretreatment? In principle, there could be two mechanisms why we have higher levels of these metabolites upon copper pretreatment. The first mechanism is that the levels of these metabolites is induced and just transgenerational retained. And the second mechanism is that these metabolites could be primed. That means its induction is faster or stronger if the same stress recurs transgeneration. So we looked at the concentration of one of these cyanidins in the different treatments and in the different pre-treatments. And not surprised, we found that the copper induced the cyanidins by about 150%. That was expected. When we then compare the control to the copper pre-treated one in each environment, we found that in each environment we had about 5% higher levels of these cyanidins. Yeah, this is not a big difference, 5%. However, I would like to mention this is certainly significant, and we are measuring these metabolites at the end of the fitness assay, meaning we're about 8 generations away from the initial stress. So yes, the cyanidins are induced and then likely simply transgenerational. We were then wondering whether it's really cyanidins that may protect photosystem 2. To address this, we simply correlated the concentrations of these cyanidins with the FVFM value, so how well the photosystem 2 is working. We found that under control conditions, there was actually no correlation of the cyanidin concentration and the FVFM value. However, under copper excess, we found a positive correlation between the levels of these anthocyanins, and FEF. Meaning that likely, yes, the cyanidins protect photosystem 2 under complex. This would mean that cyanidins likely also benefit plants. And to confirm this, we again correlated the cyanidin levels to the growth rate of the plants and found that under control conditions, there was a strong negative correlation of cyanidins to the growth rate, indicating that there are certain costs involved in the production of these metabolites. Under top access, we still had a negative correlation, but this negative correlation was not as strong, so not as steep, indicating that the costs of cyanidin production are outweighed under top access, so that cyanidins protect the plants against complex. So taken together, this would indicate that under top access, the anthocyanins are induced, and also across 5 generations of growth of control conditions, these metabolites are retained to a certain level, and this likely is costly for the plants and reduces plant growth because the production of these metabolites need to be processed. If, however, the same stress recur, we have still these retained levels of the metabolites, and this likely benefit. We were then wondering whether we also have this transgenerational plasticity and whether this transgenerational plasticity is relevant if another stress recurs. So not crop access here, but our water lily. Because in nature, often the water lily aphids invade duckweed populations in late summer, so these populations likely experience other stresses. So meaning we did the very same assay, however, only 6 genotypes, and he had induction with pre-treatment phases, control on copper, but then during the fitness assay had 1 additional treatment where we grew the plants in the presence of the aphid. So we first wanted to know whether we have this transgenerational stress resistance upon elicitation by crop access. So we look at the pre-treatment ratio of growth rate under the different environments here of the fitness assay with the different plant genotypes. We found again under control conditions the pre-treatment did not play a big role. However, under crop access, again, we had a very genotypic specific variation in its growth rate, and the same happened when we grew the plants under aphid. We were then wondering whether this copper pre-treatment not affected the plant growth, but also the growth of the aphids. So we again did the very same, and this very same assay assessed the aphid growth rate in the plants that had been previously pre-treated either with copper or control conditions. And to our surprise, we found that across these genotypes, we had about 10% higher growth of the aphids. And this is maybe again not such a large effect, 10%. However, this is really 10% in thickness of the growth of the aphids. And I also would like to highlight that this is an average of these 6 genotypes. We also had some genotypes that supported about 50% higher growth of these aphids. So yes, copper pre-treatment not only affects the plant fitness, but also boosts the aphid growth. So we were wondering, so how come? So which metabolites could account for this increased aphid growth? So again, we did the screening of our metabolites. We looked at which metabolites are increased or reduced. Under control conditions, we again found the cyanidins that were transgenerationally retained. When we, however, looked at the metabolites under aphid herbivory, there was one metabolite that popped up and caught our attention. It was OPDA. So OPDA, you may know, is a precursor of the jasmonate pathway. It itself has hormonal activity, but it is also converted into jasmonate, one of the major defense hormones against it. And that was really funny because we had this increased aphids growth, but also increased OPDA. So we wanted to look into OPDA into more detail. We first asked whether OPDA is induced and retained or whether it is. So this is a little bit a more complex figure, but I will guide you through. We have here the levels of OPDA, and we have the different pre-treatments and the different pre-treatments here and treatments. So we found that OPDA was slightly induced by about 50% when we exposed the plants to carbon. And these induced levels were not really retained when we grew the plants for 5 generations under control conditions. So we had about 30% higher levels of the copper pre-treated compared to control pre-treated plants. However, these differences were not significant. When we then, however, looked at the OPDA levels between the copper and control pre-treated plants and the aphid herbivory, we found that OPDA levels increased by about 90%. So we see that we have a pre-treatment effect of this copper pre-treatment on the levels of OPDA. Copper pre-treatment increases OPDA, particularly under aphid herbivory. So this OPDA is mostly prime, a transgeneration by initial exposure to the plants to copper excess and then the exposure of the plants to aphid. So that means when we expose the plants to copper excess, not only anthocyanins are induced and then retained, but also jasmonates are induced. They're not really retained, but they're again transgenerational prime upon exposure to the aphid herbivore. This is what we know. We also know that the aphids grew better upon previous exposure to coppers. And this is funny and made us wonder whether it's these increased levels of jasmonate that caused the boost in aphid. We were also wondering whether we see this priming of the jasmonate, which felt a bit funny to us, also when we trigger the transgeneration plasticity with aphids and not with the copper. So we first wanted to assess the first question, whether jasmonates are primed and boost aphid growth when there's initial stress on the aphid herbivore. So we did a very similar assay, but this time we did not elicit the plants with copper, but with aphids, and then measured plant fitness and phenotype under control conditions in the aphid herbivory using 8 plant genomes. We first assessed plant growth, and here we're now not looking at pre-treatment ratios, but just at actual growth rates under control conditions in aphid herbivory of the different pre-treated plants. We found that under control conditions, the aphid pre-treatment had a slight positive effect of plant fitness. However, under aphid herbivory, the plants actually suffered from the aphid pre-treatment. So they had here about 10% reduced plant growth rates. So the plants do not defend better when they're pre-treated with aphids. No, in contrast, the aphid pre-treatment harms plant growth under recurrent aphid herbivory. This also indicated that if we have reduced growth here, the plants will likely have increased aphid growth, which can cause this reduced growth here. So we measured therefore also aphid growth and compared aphid growth here between the control and aphid pre-treated plants, and found that these aphids grew about 5% better when the plants before 5 generations ago were pre-treated with aphids. So again, here 5%, not such a big difference. However, again, we have genotypic variation, so some genotype had about 10 to 20% higher growth of the aphids when they were pre-treated with aphids 5 generations. So yes, the aphids benefit from previous aphid herbivory. So we were wondering why. So which metabolites could it be? Is it really JA that plays a role? So therefore, we again measured our metabolites in a single plant genotype that showed intermediate levels of aphid growth and plant growth, and assessed these metabolites. So under control conditions, we then had some amines that popped up and some other metabolites that may be defensive. Under aphids herbivory, however, there were many more metabolites, and there was one metabolite again that caught our attention. Of course, this was jasmine. So we had jasminic acid that was about 70% higher in levels of the aphid pre-treated plants compared to controlled pre-treated plants when we exposed the plants to the aphid. So we therefore asked again the very same thing. It was jasmonate inducing, retained, or transgenerational prior. So we're looking here at the JA levels of the different pre-treatments and of the different treatments. So we found that when we exposed the plant to the aphid herbivore, jasmonates were not induced. So we actually had a 20% lower levels of this jasmonic acid. However, this difference was not significant. So not much. However, when we then compared the plants under aphid herbivory that were either pre-treated or not pre-treated with the aphid herbivory, we found that the previously aphid-exposed plants had about 70% higher levels of jasmonates compared to the plants that had never experienced aphid herbivory. So here clearly the jasmonates were transgenerational. So that made us really wonder, because jasmonates are classical defense hormones, so they should actually suppress aphid growth. However, we had increased aphid growth. So we were wondering, what does JA do to the growth of the aphids? To answer this, we did an induction experiment. We externally applied methyl jasmonate to these plants for 72 hours and then assessed aphid growth across 48 hours. And then measured the growth rate of these aphids between these two induction treatments. And we found, just very consistent with our data from the transgenerational experiment, that the plants treated with methyl jasmonate had higher growth of the aphids, had higher growth than the control treated plant. So I was really not sure whether this is true, or we were just a bit lucky when we did the experiment. So we did this experiment twice again with different students and with different plant genotypes, and we always found the same. So I'm quite confident to say that yes, jasmonate boosts aphid growth in houses. So this means that when the plant is exposed to the aphid herbivore, the jasmonates, they're actually not induced, they are maximally suppressed, but rather not changing these levels. But if we then grow the plants for few generations under control conditions and re-expose the plants to the aphid herbivore, these jasmonates were positively primed, and this likely boosted the aphid growth and suppressed the growth. So together these data show that plant defenses are not only plastic within a generation, but they can be plastic also across generations. Particularly in our case, jasmonates and anthocyanins, the transgenerational plastic, and likely altered plant and aphid. We should, however, be aware this transgenerational plasticity is not only beneficial for the plant. We saw now 2 cases in which transgenerational plasticity actually harmed plant fitness, but instead boosted the growth of it. So with this, I would like to thank the people involved in this project. So the work has been really done by Alexandra Chavez, an outstanding PhD student in my lab, who was really driving this project. And we would like to thank Martin Schäfer for the metabolomic and metabolite analysis, as well as Lisa here for her contributions. I also would like to thank our collaborators and, of course, the funding agencies. And the last announcement that another PI in Mainz is searching for PhD and postdocs in inductive genomics and evolution phytochemistry. So if you're interested, please contact. So thank you very much for your attention, and I'm looking forward to your questions.
Thank you very much. That was a very impressive presentation. An awesome system to work with. Pretty neat. Yeah, do we have any questions from the audience? Please free to raise your hand or put the questions in the chat and I can read them. Everything is possible.
Hello.
Hi, Guillermo.
Hi, Meret. Nice to see you. Nice to see you virtually, at least after such a long time. Great talks. Thank you. I really enjoy it.
Thank you.
Yeah, I just want to comment a little bit as a Jazz Monet person. So I totally buy the results. For me, are actually totally normal. So what I know, I'm not a plant guy per se or insect guy, but the what I understood through the years working with aphids and so on, I think the feeding habits matter here. They are not very good per se at inducing jasmonates. So the fact that somehow they get induced is fine. And what I want to tell you is that probably so ask you is whether you measure sugar. So because I think sugars is a big problem for aphids. So the honeydews, they always want to get rid of excess of sugar, and jasmonates are known to kind of relent at least the primary metabolism in that regard. So if you see this kind of defenses going up, sugar going down, so it makes sense. I think what I recall from the group in Cornell from George is that I think in May, so they see something like that, pretty consistent. So that when you get the jasmine induction, the aphids do better actually, because probably they have other sources or not that many sugars at least. So yeah, the question is goes around this, the sugar content and things like that, whether you check that.
Yeah, so that we want to measure sugars, and I think we still have the sample, something on our to-do list. You're totally right. I think that would be good to measure. Yes, we did measure amino acids to test whether the plants could be more nutritional, and there we did not find much changes. But it's always a question also when we grind up the plant. I mean, how relevant is it what the aphids really experience? So that's a little bit tricky, but I think the sugars would be certainly a good thing to measure in this. I agree.
Awesome. Very nice work.
Yeah, are there any other questions? Alex?
Yes. So forgive me if you mentioned this, but did you look at the paired effects of individual genotypes across the experiments? So you were looking at these aggregate trends. Were there some particular genotypes that really weren't affected by any of the experiments? Or was it random? Or was it that certain genotypes were more responsive, just in general?
So in general, when we wanted to know why is there genotypic variation on the first place. And we found that the genotypes that had increased resistance under copper access under so by when they were elicited by copper and re-exposed to copper, so those genotypes that benefited from previous copper exposure had reduced growth under control conditions and vice versa. So this is on the one hand. There was, however, no cross resistance. So when we plant or a genotype that was resistant under recurrent copper excess, this was not more resistance or more susceptible under other stresses. We looked at aphids, we looked at salt, we looked at dicot, which is a herbicide. So there was no sign of cross resistance or cross susceptibility at all. But these genotypic effects, they were reproducible. So we did about 16 genotypes and they behaved in the same way.
Thank you. Great talk.
I also do have one question. I was wondering, do these duckweeds encounter copper stress in nature? Is it more like an artificial stressor and
I think it most likely became more and more important recently because of the pesticide, but also because of runoffs from pipes. And so the anthropogenic sources are the majority at the moment. Of course, there's also some natural sources. These are likely more stable. But the anthropogenic sources, they likely fluctuated during the season. Because also duckweeds, they grow in small water bodies usually. And then we have a lot of exchange of water quite quickly. So the concentrations of copper may really fluctuate. But it's really, it's more anthropogenic than natural. I mean, natural also can occur if you have a copper source. It can occur, but may not be as common as now.
okay, thank you. Luke, please.
Thanks. Yeah, thanks for a nice talk. This is so interesting. And the black slide backgrounds are a lot of work to create the figures. Nice work. I'm curious about the mechanism of the induced compounds that are providing resistance to copper. Are these chelators, or do we know how these are working? And I'm sorry if I had to step out for a brief moment during your talk, so I'm sorry if you mentioned it.
Yes, so no, I didn't mention. So the flavonoids and the anthocyanins, they're antioxidants. How exactly they are antioxidant, I'm actually not completely sure.
I see, but this is, I see, not directly.
That is certainly known. That is certainly known.
Okay, I see. But this is not, it seems less likely to be directly impacting the copper itself and more so the process by which the copper is damaging the plant, perhaps.
I don't think that he ate him. Yeah. But not 100% sure.
But I will have a look. Thank you again.
Dirk, please.
All right. Thank you, Meret. It was a great talk. Very much enjoyed it. So the duckweed system, do you have genomic resources to look into the molecular genomic mechanisms for this transgenerational plasticity?
Yes. So we have about 200 genotypes, and all of those genotypes are sequenced, resequenced the genomes. And we wanted to do a GWAS on these for this transgenerational stress resistance. That's why we started with these 56 genotypes. It was our first batch, but then Corona hit in, and we never picked up on this approach. But I still have it in mind to continue on. But it's quite a bit of work to feed 200 genotypes. And we have a strong linkage disequilibrium. So I'm not perfectly sure whether duckweed is a good system for forward genetics. Yeah.
Oh yeah, all right, right. I can see you shared that. Great, thank you. Fantastic.
I do have one last question. I just don't know. I was wondering, do these duckweeds, or maybe you mentioned that, do they ever reproduce sexually, or is it just vegetatively?
The Spirodela, we never saw it reproducing sexually. We can make it produce flowers. We cannot make it produce seeds. And I've never heard of anybody who can or has ever seen a seed of Spirodela. However, if you look at it genetically, so if you look at the linkage disequilibrium, we see that in the North American populations, the LD is huge. So there's very likely very little sexual reproduction ongoing. However, in the Southeast Asian population, the LD is much shorter. So there still seems to be current or at least recent sexual reproduction in these species.
Okay, that's quite interesting.
There is one other genotype from which we can get seeds, but it is very rare in duckweeds that one can get seeds.
Thank you. Yeah, just one last question. Do people still have, or like one last point, do people still have questions? If not, please raise your hand now. If not, yeah, I would like to thank Meret again for this wonderful presentation and for your time and for accepting our invitation. And I hope to see everyone in 2 weeks for our last presentation of this season, and then again in January for our first presentation of the new year. Thank you very much.
Very much and giving me the opportunity to talk.
You're welcome.
Thank you,
Very
Meret.
welcome.
Thank you, guys. See ya.
Okay. Goodbye.